In this video I'll be talking about ribosomal RNA, rRNA, as well as ribosomes, and ribosomal RNA genes, otherwise known as rDNA. Ribosomal genes are transcribed as an operon, so more than one gene is included in this. So there's a small subunit gene, large subunit gene, and 5S in bacteria and archaea, and in eukaryotic genes, there's a small subunit gene, a 5.8S gene, and a large subunit gene. The 5S is often in a different location, and in eukaryotes, the major transcript is transcribed by RNA polymerase 1, or POL1, and the 5S gene is transcribed by polymerase 3. In bacteria and archaea, they have only one polymerase, so that transcribes the entire operon. It's specifically transcribed using different promoters than those that are found in tRNA and mRNA genes. You can also see a difference in some of the spacers. So in the bacterial, archaeal, and eukaryotic ribosomal RNA genes, they have a similar 5' region that contains a promoter. And then they have internal transcribed spacers. So these are transcribed as RNA and then later chopped out. Bacteria and archaea have only one internal transcribed spacer, and sometimes one or more tRNA genes are located within the spacer also. The spacers in eukaryotic ribosomal RNA genes, they differ in that there are two internal transcribed spacers, ITS1, which corresponds to the internal spacer in bacteria, and ITS2 that has more similarities to an intron. And if you look at the 5.8S gene, it corresponds to the 5' region of the large subunit gene in bacteria. So it's been split by what looks like an intron in the case of eukaryotic ribosomal RNA genes. Then they both have a spacer at the end, and bacteria and archaeal also have their 5S ribosomal RNAs in that place. And sometimes they have tRNAs located in that final spacer also, that external transcribed region. The small subunit ribosomal RNAs are incorporated into the small ribosomal subunit, and they act to attach the mRNA to the ribosome as well as ratchet the mRNA through the ribosome 3 nucleotides at a time that matches up with the anticodons on the tRNAs during translation. The small subunit RNAs are between 14 and 19S. The larger ones are in eukaryotes, and the smaller ones are in bacteria and archaea and organelles. The large subunit ribosomal RNAs are within the ribosomal large subunit, and these hold the catalytic region, the peptidyl transferase, that adds one amino acid to another in the growing polypeptide chain. And these range in size from 21S to 29S. The smaller ones in bacteria and archaea, and the larger ones are in eukaryotes. And the S stands for Svedberg units, and that's a measure of how far they will go through a sucrose gradient. So the larger ones wind up further down in the gradient, and the smaller ones wind up higher in the gradient, and so those are called Svedberg units, and those correspond to the lengths of those RNAs. In all organisms, the ribosomal RNAs are heavily transcribed and usually constitutively. That means constantly. And in the case of eukaryotes, they're even transcribed during mitosis. So even on the condensed chromosomes, that region is still transcribing, while other regions have turned off. If you look in the electron microscope, you can find this kind of structure, And this indicates the DNA running down the center. This is the start of the gene here and the end of the gene there. And these are all the transcripts coming off of that. So you can see these RNA polymerase molecules are lined up, one next to the other as they're transcribing. So it's heavily transcribed and nearly constantly transcribed. In eukaryotes, you also have this region up here called the intergenic spacer, where you find repetitive elements which attach more RNA polymerase molecules, Pol1, to those regions so that they're ready to pop onto the promoter when that region becomes open again. RNA polymerase can transcribe RNA at a rate of about 50 nucleotides per second. So per second it would be 50 nucleotides if only one RNA polymerase molecule was transcribing the gene. But if you load it up with 100, then you can increase that rate to 5,000 nucleotides per second, which is quite a bit higher. And this rate is needed in order to make enough ribosomes for each cell prior to cell division. In bacteria, it depends on the bacterium, there are from usually 2 to 10 copies of ribosomal RNA around the genome, sometimes as low as 1. And they're dispersed, so they're not all together on the chromosome. And after they're transcribed as an operon, as I said, they have to be processed. So if you look at the processing, usually the 5' end is cut off first, and then the 2 subunits are split, and then those spacer regions on the 5' and 3' end are then processed off and finally incorporated into the small and large ribosomal subunits, including the proteins that are needed to hold the RNA into its functional form. The proteins do little else than to just hang on to the RNA to make sure that it's in the form needed for a functional ribosome. In the majority of eukaryotes, more than one copy is found along the chromosome in tandem arrays. So you have one transcription unit here, one gene, and one containing the large and small subunits and 5.8S genes. Another one here, and you can have hundreds of these in a row. And each one of those, of course, as I've already explained, consists of an intergenic spacer between the 2 copies, tandem copies, a promoter, and then the gene, which is transcribed as one long transcript and then processed into the 18S or small subunit, 28S or large subunit, and 5.8S ribosomal RNAs to be incorporated into the large and small subunits. The 18S becomes incorporated into the small ribosomal subunit, and the 28 and 5.8S ribosomal RNAs end up in the large ribosomal subunit. Here's one example of how they are arranged in most eukaryotes. This is one from Vishwafaba or broadbean. This is one I worked on for my PhD. On chromosome 1, it's a very large chromosome. There's a centromere, and then there's a secondary constriction. And often you find ribosomal genes located at the secondary constrictions on chromosomes. And it's actually a misnomer because it's a region that's not constricted at all. The other regions of the chromosome are constricted in preparation for mitosis. The ribosomal region is still looped out and transcribing during mitosis. So although it doesn't stain well, which leads to people thinking it was constriction, it's actually a region that is not constricted. If you pull out the ribosomal RNA gene region, it looks like this, where you have a transcription unit here with the small subunit, large subunit, and 5.8S gene, and then an intergenic spacer, and then another transcription unit, and another IGS, and another transcription unit, etc. And so if you focus on just one of those repetitive regions, you find, or transcription or transcribed regions, you find this where there's a small subunit gene, large subunit gene with the promoter. And these regions down here, which are subrepeats. And each one of those in Vishafaba, anyway, is 325 base pairs in length. And those are regions that have similarities to the promoters. So this is where those RNA polymerase POL1s can be concentrated so that they're ready to pop onto the promoter when that region becomes free as the next polymerase has started to transcribe that gene. And again, this is transcribed as one large piece, one large RNA, usually around 35S, so it's a very large ribosomal RNA precursor. And then that's processed into the small subunit, the large subunit, and the 5.8S ribosomal RNA, and put together into the small subunit of the ribosome and the large subunit of the ribosome. And as I indicated before, the transcription rate is about 50 nucleotides per second, sometimes faster. And then if you load that region up with polymerase molecules, you can increase that by 100. And then if you have 100 copies of those, as most organisms, most eukaryotes do, you can get the transcription rate up to 500,000 nucleotides per second. Here's the processing of the ribosomal RNA in a eukaryote, and you can see that it's processed in a very sequential manner. So here's the gene up here, and then the transcript, which is in this case a 35S transcript. I think this was from work with yeast. The first thing that happens is that end that contains the promoter region is chopped off, and then that 5' external transcribed spacer is cut off, and then the ITS1 is chopped in, and so that separates the 20S, which contains the small subunit gene, or the small subunit ribosomal RNA, from the piece that contains the large ribosomal RNA and the 5.8S ribosomal RNA as well. On the small subunit side here, then the 20S, that little piece on the 3' end of the RNA is chopped off, and that becomes the 18S, or small subunit ribosomal RNA, that then becomes the major functional piece of the small ribosomal subunit. On the other side, you can get a couple of different types of processing. A common one is here in the middle, where the ITS2 is chopped, and then you have 7SL and eventually the 25S, or large ribosomal RNA, and the 5.8S ribosomal RNA. In some cases, the 5.8S is a little longer, so it becomes a 7SL. It has a few additional nucleotides on the 5' end of the RNA. And then that's processed into the large subunit here, which is the 25S in this case, and the 5.8SL, the slightly longer piece of the 5.8S gene. In both cases, the large ribosomal RNA and the 5.8S ribosomal RNA are put together into the large ribosomal subunit, which becomes the catalytic part of the ribosome. Because bacteria and archaea don't have a separation between the where the DNA sits and the cytosol, or the cytoplasm, these things are occurring in the same compartment. So you have the ribosomal RNA genes down here being transcribed, put together into ribosomes, and then the ribosomes come together onto the mRNAs, which are being produced in the same area, and so those immediately attach to the mRNA and start translating those messages into proteins. In eukaryotes, those two functions are separated because the DNA and assembly of the ribosomes occurs in the nucleus, and translation occurs out in the cytosol or on the rough ER. This complicates the process a little bit because then you have the transcription of the ribosomal RNA here. You also have transcription of mRNA genes here, but they can only get together once they're outside of the nucleus, so that then you have translation ongoing out here. And then if the proteins such as RNA polymerase and things like that are needed, they have to be imported back into the nucleus. If they're winding up out here, then they either wind up in the cytosol or in the rough ER, where they're incorporated into organelles or onto membranes. The construction and assembly of the ribosomes is also a bit more complicated because, as I said, that occurs in the nucleus, and it actually occurs in a region of the nucleus called the nucleolus, where you have the transcription of the ribosomal RNA, and then assembly of that RNA using the ribosomal proteins, and then those are assembled into the small subunit and large subunit, shipped out or exported out of the nucleus, and then they attach to an mRNA out there and start translating the proteins. And those proteins, which are needed for the ribosomes, then are translated out here in the cytosol, and they are then transported back into, or they are transported into the nucleus and into the nucleolar region to then assemble into the ribosomes. The 5S ribosomal RNAs are actually transcribed in the nucleus in other locations, not in the nucleolus, and then they're shipped over into the nucleolus for assembly into the large ribosomal subunit. While the small subunit doesn't have a lot of problem getting through the nuclear pore, the large subunit with all of the proteins and the ribosomal RNAs is one of the largest things that has to move through a nuclear pore. So if it were any larger, it would have difficulty even fitting through that nuclear pore. So here is an example of that, of a nucleus and a nucleolus. This is a plant cell down here with the nucleus and the heterochromatin, which are the dark areas, and the euchromatin, the lighter areas where the DNA is actually being transcribed probably. And then the nucleolus, which is clearly visible here. And you can see it doesn't have a membrane around it. And here's a close-up of the nucleolus, which contains a fibrillar region, which is where the RNA is being transcribed, and a granular portion where the ribosomes are being assembled. Both the ribosomal RNA and the ribosomal proteins are being put together into the small and large ribosomal subunits. And remember in a previous video when I talked about planktomycetes possibly being the source of a nucleus. Here's Gemata again, obscurula globus, and you can see this membrane, double membrane around here, which looks sort of like a nucleus, and then there's this darker area in here that contains the DNA, of course, but it looks, at least superficially, somewhat like a nucleolus. So another case where the similarities between a nucleus in eukaryotes and these nucleus-like structures in planktomycetes are evident and convincing. When the structures of the small subunit ribosomal RNA have been determined, all of them in bacteria look similar to this one, where you have the 5' end here going through this domain down here, and then up through this domain up here, up to this one up on the upper right, and then down into this last domain, and then there's the 3' end. Even when you look at structures in archaea and eukaryotes, you can find some of these same regions, but they're larger and much more extended in both of those, especially in eukaryotes where they've expanded quite a bit. And you can see some of these common structures that I was talking about when I introduced RNA, in that there are stems and loops, bulges, internal loops, and other structures throughout these molecules. When you compare these different domains from the secondary structure to the tertiary or three-dimensional structure, you can see that they occupy very specific regions within that molecule. So here's the first domain here in red, you can see it down here, and then the green domain over here, and then going into this yellow domain, and finally the blue domain here, which is the one that holds the mRNA on this 3' end. That's where it starts grabbing on to that mRNA molecule. And in fact, it does this by hydrogen bonding to it, by forming a duplex, because the 3' end contains what's called an anti-Shine Delgarno sequence, and mRNA in bacteria upstream from where the translation starts has a Shine Delgarno sequence. So they hybridize together, and that's what attaches the mRNA to this small subunit RNA in the small ribosomal subunit. The large subunit can also be split into several domains. Here's 1, 2, 3, 4, 5, and 6. And then here's the 5S that attaches to a part of the large ribosomal subunit also. Most of these regions are for attachment to other molecules, other RNAs and proteins. But this region in here attaches to the tRNAs, and then this loop in domain 5 right here, that contains the peptidyl transferase activity, which attaches one amino acid to another amino acid in the growing polypeptide chain. So that's the enzymatic portion right there. So here are a couple more views of the ribosomal, both the large and the small subunits. This is the secondary structure of the small subunit. It's flipped over a little bit, so this is the 3' end where you would have the anti-Shine Delgarno sequence. And in this, which is again the small subunit, the anti-Shine Delgarno sequence would be somewhere in the middle here of that one. Here is a different view showing the large subunit and small subunit with the tRNAs at the A and the P site, and then the L1 is a loop where the amino acid, the tRNA without the amino acid, is pushed off or exits the ribosome. And here's how the RNA is threaded through the small subunit, which then attaches to codon by codon to the anticodons of the tRNAs. And here, this is the small subunit and the large subunit, showing the faces that attach to one another. So this is sort of like taking a cookie and breaking it apart and looking at the inside. And so here's the tRNA at the A site, the tRNA which is holding an amino acid that will be the next amino acid added, and then the tRNA at the P site, which is holding the growing polypeptide, and then the tRNA at the E site, which is the one without an amino acid. The amino acid has been pulled off of that, and it's exiting the ribosome. Here are some views of the ribosomal RNAs in the ribosome that have been determined by X-ray crystallography. So these are, high-resolution images. And so this one at the top left shows the small subunit on top, And then the next one shows a side view of the small and large subunits. And you can see the tRNAs in there also, how they sit within that area between the small and large subunit. Here's another side view down here in the lower right, and then a bottom view from looking from the large subunit end of the ribosome. So if you look at these side views, this orange tRNA here is the one entering, bringing the new amino acid in. And this red one on the other side of the ribosome, that's the tRNA that has lost its amino acid and is coming out of the ribosome on that end. And this is again opening the ribosome up. So you can see this orange tRNA still has its amino acid on the end. The red one has the amino acid plus the other amino acids in the growing polypeptide chain on it. And then this red tRNA on the other side is the one that's leaving the ribosome, having lost its amino acid, which has been added to the polypeptide chain. And this is a more stylized version just to show you the relationship between the tRNAs, the one at the A site in red, the one at the P site in green, and the one exiting in yellow. And then how the mRNA is threaded up through being brought by the small ribosomal subunit, and then the catalytic site, the peptidyltransferase, would be in the middle here of the large ribosomal subunit. So as you know, the translation process is done with high fidelity. That is, very few, although some mistakes are made. So one of the reasons it does have high fidelity is there is a elongation factor during translation called EFTU, which is a protein that sort of checks to see whether that tRNA is going to stick in that site, meaning there's a strong reaction, hybridization essentially between the codon of the mRNA and the anticodon on the tRNA. And so it flips in and out as the tRNAs are brought into that site. And here it is shown where it's flipped down onto the tRNA that's moving into the A site of the ribosome. And here's another depiction where it's flipped out, so it's sort of on a hinge where it can flip in and out of that. So it's already checked to make sure that that tRNA at the A site is correct. The one at the P site is there, and then the peptidyltransferase function is somewhere in here, and it's adding this amino acid to this chain, and then transferring essentially that chain to this one at the A site, and then moving it over into the P site as this one is exiting. As I indicated in a previous video, this didn't happen all at once, right? So very early on, prior to bacteria, there were ribosomes in those RNA-based organisms, but they were very simple, and they rapidly became, rapidly over the course of a few 100 million years, they became more sophisticated until by about 3.5 to 3.8 billion years ago, then you had a bacterial ribosome. And it's quite a bit smaller than a eukaryotic ribosome, shown here as a protist. So the bacterial ribosome consists of a 50S large subunit and a 30S small subunit, whereas in a protist or a fungus, you have a 60S large subunit and a 40S small subunit. In archaea, they're closer to bacterial size, but they're larger and a little more sophisticated than those in bacteria. But remember from our evolutionary trees and what I've talked to you about endosymbiotic relationships and phylogenetics, that archaea were the source of the eukaryotes. So although the archaeal ribosomes are closer in size to the bacteria, certain characteristics of those ribosomes look more like eukaryotic ribosomes than bacterial ribosomes. And here's a comparison between the large ribosomal subunits and the small ribosomal subunits for bacteria on the left and eukaryotes on the right. And so you can see there have been size changes, and there are certain characteristics such as different loop outs. But in general, the location of the AP and E sites and other functions of the ribosomes, especially the central portions of the ribosomes, are very similar in the two, and they perform the same function. That is, using the peptidyl transferase function of the large subunit to link the amino acids together. There's an A, P, and E site for attachment of the tRNAs, and then the small subunit acts as a ratchet to move the mRNA through the ribosome at a rate of 3 nucleotides per amino acid added. And I will talk more about ribosome evolution in a later video. So illustrated here is translation, the process of translation in bacteria, including initiation, elongation, release, and recycling. So initially you have an mRNA, a small subunit, and some tRNAs, as well as some IFs. Those are initiation factors, which then will bring the mRNA to the small subunit. It will then attach to the small subunit at the Shine-Delgarno sequence of the mRNA. And then once that complex is together, it will attach to the large subunit, and the initiation factors will come off. And then that initial tRNA and the elongation factor, EFTU, will attach. And you'll start getting movement using GTP, that ratcheting effect of the mRNA with regard to the rest of the ribosome. And then amino acids will be brought in by tRNAs. And another elongation factor, EFG, will be brought in. Then GTP hydrolysis will occur, and that shifts the RNA using motion in the small subunit, and it'll shift the RNA by 3 nucleotides, another codon, and then go through the process again, adding amino acid after amino acid after amino acid based on the codons of the mRNA and the anticodons of the tRNAs. Once a stop codon is reached, then releasing factors, RFs, will be added, and that will then release the protein from the ribosome, and then other release factors will come in and cause a decoupling of the large and small subunits, and then they'll be recycled to attach to a new mRNA, and then start the process again for a different mRNA to decode that and produce a protein. If you compare that to translation in eukaryotes, so bacteria are on the left-hand side and eukaryotes are on the right-hand side, the process is very similar, especially the central region. And if you look at the initiation factors, elongation factors, and release factors, some of those are in common to both. So if you look over here in the eukaryotes, the ones that are listed in black are the same ones that you find in bacteria. And then the ones that are in red are unique to eukaryotes. So if you go down here with bacteria again, here's the mRNA, there's the Shine-Delgarno sequence attaching to the anti-Shine-Delgarno sequence with the help of some initiation factors to attach the mRNA to the small subunit. That then attaches to the large subunit, which is the catalytic part of the ribosome. And then this middle portion is very similar in that you have elongation factors acting here to add the amino acids one by one being delivered by the tRNAs just over and over and over repeated until you reach again a stop codon. And then you have release factors that will then pull off the mRNA, the protein, and eventually separate the two subunits. The initiation in eukaryotes is a bit different in that you don't have a Shine-Delgarno sequence, you have a Kozak sequence, which causes circularization of the mRNA, which signals that that mRNA is ready to be translated. And then you have initiation factors coming in to attach, again, the mRNA to the small subunit, usually by a cap binding protein, also recognizing that 7-methylguanosine as the attachment site in the small subunit, bringing that together with initiation factors with the catalytic subunit, that is the large ribosomal subunit. And then you get other elongation factors added to that, and a bit more complex as far as translation in the middle here, but it's very similar in both cases. And then a stop codon is reached. Release factors then cause release of the protein, and then separation of the large and small subunits from one another to start the process all over again. So now if we go back to the structure of these ribosomal genes and ribosomal RNAs, each one of these regions, then if it's mutated during evolution, has a unique effect in many cases on translation. So if you have some mutations in the external transcribed spacers, chances are they won't affect translation all that much unless they're involved in processing of the ribosomal RNA during some point. The small subunit, mutations in there can have a large effect. Mutations in the large subunit can have a large effect. And even mutations in the 5S can have a large effect. Mutations within the spacers probably have less effect, but it really depends on what the mutation is and where that mutation is in the process of creating the ribosomal RNAs and then eventually the ribosomes. So if you look at the effects of mutations for various regions in the ribosomal DNA and the ribosomal RNA genes, if you have a mutation in the promoter region, you can increase or decrease expression, which can have a negative effect on production of ribosomes and negative effect on the cells. If you have a mutation in those intergenic spacer regions, which hold the reserve RNA polymerases, those can also cause an increase and decrease in expression. And these have actually been measured experimentally. If you affect a peptidyl transferase, normally that's detrimental and in many cases lethal if you affect how an organism can put together its proteins, right? Movement of the mRNA, which is caused by GTP aided movement of the small subunit, that can also have very detrimental effects on translation and survival of the organism. If there are mutations that affect where the tRNAs fit and sit, that's also very detrimental or lethal to the organism. If you have mutations in the spacers, either the external spacers or the internal transcribed spacers, usually the changes don't affect much, but it depends on where those mutations occur. If they're closer to the ribosomal RNA genes themselves, the small subunit or large subunit, sometimes they can have major changes, major effects. Introns, there are introns in ribosomal RNA genes. They can have either no effect at all, or they can be completely lethal. So there's a big range of what can happen there. If the mutation affects the structural parts of the ribosomal RNA, so it affects the structure of the ribosome, it depends on where it occurs. If it's in the very center of the ribosome where those active sites are for tRNAs and the peptidyltransferase and movement, then they can have major effects. If they're on the outside, often they don't have major effects at all on the ribosome functionality. And something I didn't talk about, but there's a hole going through the large subunit, and that's where the growing polypeptide is moved through the ribosome as it's produced, and actually has a purpose in that it holds on to that peptide and actually has held onto that polypeptide for probably 4.2 billion years. So it's been there ever since the beginning to stabilize the reaction so that the ribosome can do its job to attach the polypeptide, the two amino acids together, or the amino acid to the growing polypeptide chain. So that hole going through the large subunit is absolutely necessary. And if anything happens to that hole, blocks it or alters it in any way, that can be detrimental or lethal. And there are actually some groups working on trying to develop drugs that would block the hole of the ribosome for fungal ribosomes or bacterial ribosomes, etc. And finally, if there's a mutation in the non-structural, non-control, non-enzymatic portions of the ribosomal RNA gene locus, often there are no apparent effects. So I go through this list primarily, and this is the first instance that I've done it for these videos and for class, to indicate that a mutation in a gene, its effects at least, are dependent on what the mutation is, a C to T change or a C to U change in the case of RNA, or an A to G change, an A to U change, something like that. Not only the mutation itself, but where it occurs in that gene. And all mutations then are not equivalent. Many are completely silent in that in some portions here, for instance, in this 5' transcribed spacer, you could have a mutation occurring, and they do occur more often, and it would have absolutely no effect on how many ribosomal RNAs are produced and how many ribosomes are produced. Whereas if you mutate a site here in the peptidyltransferase site of the large subunit, you could totally wipe out the function of the ribosome and kill that organism. Now, in the case of bacteria, that could be pretty detrimental because some bacteria only have 1 or 2 ribosomal copies. So that could greatly affect the number of functional ribosomes that are produced by that cell. In eukaryotes, often they will have hundreds of copies of ribosomal genes, ribosomal RNA genes. In that case, if you mutate one so that the peptidyltransferase site no longer works, that's only 1% of the number of genes there, and so it probably would still be able to make a sufficient number of ribosomal RNAs and thus ribosomes, so it probably wouldn't affect them as much. So a couple of different things are at work here. One is the number of copies, where those mutations occur, and what the organism is or that cell is doing at the time. So as you go back over some of this material, please think about selective pressures on ribosomal RNA, on the ribosomal DNA, in the nucleus, in the cytosol, in cells, in organisms, etc. To try and put those all, or integrate those in your mind about, what's happening to this part of the gene and how does that affect the expression of that gene for this cell or that cell, or does it affect it at all? So try to start integrating some of those things together, and we'll go through this also for some of the other types of genes as well. Okay, so that's the end of this video on ribosomal RNA, rRNA and ribosomal RNA genes and ribosomes, and their function and what mutation can do to them. In the next video, I'll talk about ribosomal gene copy number, how organisms need, or why organisms need multiple copies, and how they maintain multiple copies of ribosomal RNA genes. Okay, that's it for now. Thanks for listening and watching, and I will talk to you later.